The MCAT Podcast, session number 131. A collaboration between the medical school headquarters and Next Step Test Prep, the MCAT Podcast is here to make sure you have the information you need to succeed on your MCAT test day. We all know that the MCAT is one of the biggest hurdles you'll face as a pre-med, and we're here to give you the motivation and information that you need to know to help get you the score you deserve so you can one day call yourself a physician. Welcome to the MCAT Podcast. My name is Dr. Ryan Gray, your host here every week. And this week I am again joined by Clara from Next Step Test Prep. She is the master of content for Next Step Test Prep. So if you are taking some Next Step exams, then go check out everything. Welcome back to the MCAT Podcast. My name is Dr. Ryan Gray. If this is the first time joining us here on the MCAT Podcast, I recommend you hit pause, go back and start at the start of this journey for our Next Step full-length 10 discussion way back several months ago. So I would go back and start listening where we start with Chem Phys, Passage 1, Question 1 for Next Step full-length 10. As always, I'm joined by Clara, and we're going to jump right into our last passage here in the Biochem section. Clara, back for some more Next Step Test Prep and MCAT podcasts. I'm kind of getting excited and nervous that we're wrapping up Biochem and the unknown of PsychSoc getting into my head with PsychSoc. So I'm excited to wrap these up, but scared of the future.
Well, don't worry. There's not too much to be scared of with PsychSoc. It'll be different, but it'll be fun.
What's the data show from Next Step, the Next Step's data of how well students or how bad students are doing in PsychSoc? Is it one of the easier sections, harder sections?
Well, it's interesting because in terms of, so we have a lot of data from thousands of students that shows basically score by section and on Next Step full lengths. And then we also have collected data about official MCAT score and AAMC practice test scores. And from what we've seen, the sections are really pretty even, which makes sense, right? The MCAT is designed so that the median score in each section is the same. But what I have found are pretty big differences, like not in terms of like what score students get, but in things like timing, for example. Like you can finish a PsychSoc section in just 45 minutes, where you would never ever finish another one of the science sections that quickly. So it's different, but score-wise, not anything dramatic. Okay,
interesting. So potentially easier, but maybe because it's easier, the scores are harder to obtain.
Yes.
Okay. All right. Because they're not, what's the word? They're scaled. They're not, what's the word that everybody uses that it's wrong? I forget. Curved.
Not curved, but they are weighted.
Yeah.
Is
equated.
The
yeah. I still don't understand the difference, but hey, whatever.
Oh, it's subtle.
All right. So let's go ahead and dive into our passage today. Again, if you want to follow along, go to mcatpodcast.com and you can follow along in our show notes, download the handouts for today.
Okay, so passage 10, our last passage in Biochem. A research team conducted a number of lab experiments to investigate activity of membrane proteins in 3 different cell types. Experiment 1. Voltage clamp electrodes were used to measure the transmembrane potential difference of a squid axon and to track it as a function of time. Application of L-glutamate to the cell dendrites resulted in a depolarization of the membrane and a repolarization in the opposite direction, as shown in the accompanying chart. And then here we see Table 1. The caption for Table 1 is Transmembrane Potential Difference of Squid Axon Over Time. And if you are not following along in the handout, this is a very simple table here. It just shows transmembrane potential, and it has a bunch of different potentials starting with -70, going all the way up to +40, and then going back down to -70. And then that is mapped against time. So we see this -70 up to +40, back down to -70, all over about 5,500 milliseconds. Then experiment 2, mutated red blood cells were reproduced in a number of cell cultures, which were separated into different repositories. Repository A was placed in a 310 milliosmol per kilogram sodium solution. Repository B was placed in distilled water, and repository C was placed in a mixture which contained fluorescin transferrin complexes fluorescent concanavalin A complexes, iron 3+ ion, and a dilute concentration of vinblastine. Each repository was microscopically observed 1 hour later and 12 hours later. It was later determined that the mutation in the cell cultures affected active transport ion channels. Then the results of the environmental surroundings tests are given, and we see that culture A, after 1 hour, had no effect, culture B had partial lysis, and culture C had a slight fluorescence of cell cytoplasms. And then they also tell us after 12 hours, culture A had partial shrinkage of the cell size, culture B now had complete lysis, and culture C had increased fluorescence of cell cytoplasms. Finally, experiment 3. An extraction from a localized site of a biological specimen which was experiencing an infection due to a prokaryotic pathogen was cryofixated and imaged with electron microscopy. The resulting image depicts cells, and actually the passage did not give an image, so it must be inferred results, depicts cells identified by the researchers as neutrophil granulocytes. Several of these cells appear to have been in various stages of engulfing pathogen cells. The image additionally depicts cells identified as macrophages, which appeared to have been in various stages of engulfing neutrophil granulocytes.
C, move on.
Yeah, there's a lot going on, but the nice thing is it's really clearly divided into experiments, so some question might just ask about experiment 1, and then we know where to go.
All right. Wow. Okay. So question 53. Which transmembrane protein is most likely to have been involved with the repolarization of the transmembrane potential difference in experiment 1? So again, just experiment 1, which is nice. A, sodium-potassium passive transport channel. B, sodium-potassium active transport channel. C, sodium-potassium ligand-gated channel. Or D, none of the above. So it's interesting. It's one of those that I remember from experiment 2, at the very end, it said it affected active transport ion channel. So that almost is like triggering me to go, ooh, active transport channel. But it's referring to experiment 1. And so I'm not sure if experiment 2 is the same, if I have to keep that in mind with what's going on. But because experiment 1 doesn't really tell me anything about active or passive or ligand gated, oh man, I think I would, for time purposes, I would pick the active transport one and then move on, thinking that maybe there's some similarity with the experiments and that would be it. And then I would move on.
Yeah, it works out actually for this. So you got it. But it's interesting. It actually is for a different reason why active transport is the answer. So experiment 2 did mention that this mutation affected active transport ion channels. Mm-hmm. But the real reason why the answer to this question is active transport is because it's actually really a content question. So it's almost a pseudo-discrete, which is kind of weird because it references an experiment in the passage. Mm-hmm. But the question talks about repolarization. And when we're talking about repolarization, basically reestablishing the resting membrane potential, then that is always going to be the sodium-potassium pump. So the sodium-potassium ATPase is an active transport channel, and that is what establishes our resting membrane potential back at that -70 value.
Wow. So yeah. Good guess.
Indeed.
Okay. So, and it's one of those, I think, that kind of makes me mad as a student going through this. Like, if you're going to give me a passage, give me a passage question. Don't hide pseudo-discretes in passage questions.
That is the trickiness of the MCAT, right? Is that it's hard for this one to even see that it's a pseudo-discrete. So it is frustrating for sure.
Yeah. Okay. 54.
Right. 54. In experiment 2, what best explains the consequences of the 310 milliosmol per kilogram sodium Na+ solution on the cells in repository A? Answer choice A, partial functioning of active transport ion channels. B, biconcave nature of red blood cell membrane, decreasing the cell's structural rigidity. C, passive flow of ions through transmembrane proteins. or D, outflow of water molecules through aquaporins?
Ooh. So the repository A placed in the 310 milliosmol solution, and then the cells. So then we have the different cultures, but I'm looking at the table 2 as the different cultures, and I'm looking at the question. So I'm trying to go step 1. What is the question asking me? And I'm trying to figure out if in the experiments, culture A, B, and C, if that has anything to do with the question or not, or just the cells in general. I almost wonder if this is like another pseudodiscrete where it's saying, hey, what's going to happen when you place cells in this, I'm assuming is a hypertonic solution? And so am I on the right track of what it's trying to ask me?
You are.
Okay. So what's going to happen to the cells in a hyperosmotic solution? And we're told that the repository A is red blood cells. And so we have answer choice A, partial functioning of active transport ion channels. I don't know what that means at this point for a hyperosmotic solution. B, biconcave nature of red blood cells. So it's like, hey, red blood cells are biconcave. So it's like, oh yeah, that must be it, because it's telling me a fact. But I don't know if it has to answer the question here. And affecting the cell's structural rigidity. C, passive flow of ions through transmembrane proteins. Or D, outflow of water molecules through aquaporins. So you're placed in a hyperosmotic solution. You would think because the diffusion and osmosis and all that fun stuff, whatever is needed here, it's going to try to go from low to high to make everything balanced. So I would pick D because it seems the most logical, but I'm not sure.
D is perfect.
All right. Look at
that. On a roll.
Okay.
Yeah. This is actually a really good question, I think. And you were mostly right about it being pseudo-discrete. Basically, you can answer this question using just content knowledge. If you do think, like 310 milliosmol, that sounds kind of high, so that must be hypertonic. And then what happens when a cell is in a hypertonic solution is water will want to rebalance. So water will move from an area where there is low solute, like inside the cell, to an area of high solute. And that is exactly why our water molecules will flow out through aquaporins, which is D. So you totally can actually get it without using the passage. The thing is, you also can use the passage. Like, for example, if you didn't know that 310 milliosmoles per kilogram is hypertonic, because you don't really have to know like sort of detailed medical pieces of information like that. What you could do is you could go back to table 2, and that's where it told us that after 12 hours, culture A showed partial shrinkage of cell size. So that's where it's like, okay, we've confirmed that water must have been flowing out of the cell. That totally fits in with D.
Okay.
Yeah.
All right.
All right. Cool. And yeah, the other ones were way off. They were all kind of crazy.
Yeah.
The red blood cell membrane has nothing to do with this.
Yeah. Okay. Yeah. Question 55. Why did the lysis in experiment 2 occur? And it's a Roman numeral 1. So we have Roman numeral 1, lack of ability of transmembrane channels to relieve osmotic pressure. 2, hydrophilic nature of cell membrane working in combination with surrounding water. And 3, flow of distilled water into the cells. So we have answer choice A is 1 and 2, answer choice B 1 and 3, answer choice C 2 and 3, and answer choice D 1, 2, and 3. So this is interesting. 3 right off the bat jumps out at me as completely opposite of the answer that we just picked for question 54. Water going into the cells, because we just said water is going out of the cells from area of low solute to high solute, as you said. So I would automatically get rid of 3, which only leaves me A, answer choice 1 and 2 only.
Well, This is a funny question because experiment 2 had 3 different cell cultures. So culture A is the one we talked about in the previous question, and that's the one where water flowed out of the cell. Mm-hmm. But there were 2 other cultures, and actually the question here says, why did the lysis in experiment 2 occur? And if we go back to experiment 2, we can see that complete lysis, like the only place the word lysis shows up, is with culture B. So it's actually asking about a totally different cell culture than the question before.
Oh, interesting.
Got to be careful because, you didn't, it's an easy mistake to make because we know experiment 2, right experiment, but wrong culture. So actually 3 is a perfectly valid answer here because the opposite thing is happening than was happening before. We see lysis. Lysis usually means the cell essentially like explodes, and that can totally happen, when distilled water floats into the cells. So 3 is an answer. Yeah. And then
and then 1 makes sense for that too, the transmembrane channels to relieve the osmotic pressure so it explodes.
Exactly.
And so the question then is if 2 is correct, because if 2 is correct, then it's D. If 2 is not correct, then it's B. So hydrophilic nature of cell membrane working in combination with surrounding water. And I don't remember cell membranes if they're hydrophilic or hydrophobic. I think they're hydrophilic on the outside, right?
They are.
Hydrophobic on the inner membrane.
Totally.
Okay. And so the question is, does that have any effect on the lysis? And it doesn't make sense that it would, so I would probably exclude it and just go with B, 1 and 3 only.
Awesome. That's perfect. Yeah. Yeah, it's interesting because if we know, like the outside of the cell membrane is hydrophilic, the inner part with the phospholipid tails is hydrophobic. So the hydrophilic part isn't wrong that it touches the outside water, but it just has nothing to do with this because like the cell membrane's always hydrophilic on the outside and cells aren't always lysing.
That'd be fun. We'd have a problem if that were true.
Yeah. So that makes it a little bit easier if we just think of common sense. And then, our answer is B.
Okay. Very cool.
Awesome. All right. Last one. 56. One effect that vinblastine has been shown to have is to induce endocytotic activity in red blood cells. With this in mind, what would be the likely effect if vinblastine levels were increased and experiment 2 were to be rerun with a higher vinblastine concentration in the mixture surrounding cell culture C? So it's a long one. A says fluorescence of the cell cytoplasms would occur more rapidly due to increased uptake of fluorescent-bonded molecule complexes. B says fluorescence of the cell cytoplasms would not occur due to increased endocytosis causing more rapid evacuation of fluorescent material. C says no effect as endocytotic activity is not specific to transferrin or concanavalin A, but rather has equal effects on various types of surrounding molecules. And then finally, D says no effect as the increased fluorescence is being caused by the defect in active transport membrane proteins, which prevents them from evacuating fluorescent materials.
Like I said at the very beginning, C and move on. This one, for me, with the way that I read and my poor reading comprehension, I'd have to read this question like 3 or 4 or 5 times to figure out what it's asking me. And then each of the answers are so long that I would be scared off by this and probably just move on. But since we're on a podcast and we're not actually sitting down for the real test, we can try to think through this one. So the question is saying, what's going to happen if we add more vinblastine and experiment 2 is rerun with the higher vinblastine concentration for cell culture C? So now we've had a question about A, one about B, and now we are on C. And so just to remind the listener, culture C, after 1 hour, there was a slight fluorescence of cell cytoplasms, and after 12 hours, increased fluorescence of cell cytoplasms. And so the question is, what's going to happen with more vinblastine? It's inducing endocytic activity in red blood cells. So if it's telling me it's inducing activity, I'm going to assume that we're going to get more fluorescence because there's more activity going on. It's going to pick up stuff more. So based on that, I would probably go with answer choice A because that's the only one that says the fluorescence would occur more rapidly. And so that's what I would pick.
That's a good pick.
Oh, okay.
That happens to work out this time. I mean, it's interesting, right? Because it's fair, because we're thinking endocytosis, it'll pick up more stuff, and so there'll be more activity. And that actually is like exactly what's going on here. But sometimes you can get weird questions where they'll trick you, right? And it's like increased activity, but it's like increased activity of something that they said earlier was like an inhibitor. So it's good to be careful. But yeah, if we don't want to spend too much time on this, we can go with A. And A is perfect because we're inducing endocytotic activity. Endocytosis means essentially bringing material into the cell. And they told us in the passage that the cell culture C was placed in a mixture containing all of these basically fluorescent complexes. So if now we do more endocytosis, well, the cell was floating around with all these fluorescent complexes outside of it. Now it's endocytosing them and it's bringing more fluorescence into the cell. And then A is just perfect.
Okay.
Yeah.
Okay. So this was a pretty hard passage. For somebody who's reading the passage and like I did, kind of freaks out at the very beginning, like C, move on. What would you recommend that student do just to give them a fighting chance of maybe getting one or two right? Yeah.
Well, it's interesting, right? Because you thought this was going to be a really crazy passage, and then I think you got them all right. Or I guess there was one that was the Roman numeral one. But I would say first, people are really bad at accurately predicting the difficulty of a passage just by looking at it. Mm-hmm. Like everyone is just really bad at it. And that's one of the reasons why triaging passages doesn't help. So the first thing I'd say is just don't necessarily assume the questions are going to be really hard just because the passage is really dense.
What do you mean by triaging passages?
Triaging was a strategy that was used by some of the other test prep companies for the old, like the pre-2015 MCAT, where basically they would say, oh, the first thing you should do when you get to a new section is like click through really quickly and try to see which passages look hard and which ones look easy. Okay. And then do the easy ones first, and then maybe there's some hard ones. If you don't have time, you won't get to those. And it was a horrible failure of a method because, first of all, people would think something looks hard, and it turns out the questions were really easy, and then they would miss out. And then also it just doesn't work anymore because now they show one question at a time, so it just takes forever to click through.
Okay.
Yeah. So that's the first thing is don't assume it'll be too hard. And then the second thing is even if you just focus on getting like a little mental summary of the three experiments where you're just like, oh, one was sort of just like membrane potential and some like neuron essentially might be like content based. 2 was all these different cell cultures. And then 3 was something to do with like neutrophils. If you just even get a mental summary of all 3 of them, then it can make it feel a lot more manageable because you realize it's really just about 3 different experiments. It's not like you don't get distracted by all the dense like concatenavolin, random words. Mm. Yeah. And if you have to, you always can just pick an answer, mark it or flag it, and then move on because, yeah, if you have time at the end, great. If you don't, then probably that wasn't what you should have been spending your time on anyway.
All right, there you have it. Again, next step, test prep, full length 10, Biochem, our last passage of Biochem. Next week, we have one more set of discretes for Biochem before we move on to Psych Soc. If you are looking for a self-paced online course for the MCAT, maybe one-on-one tutoring isn't something you're interested in, maybe you don't have the budget for one-on-one tutoring, check out Next Step Test Prep and the course that they have to offer. The MCAT course comes with over 100 hours of videos, access to all of their materials, access to the AAMC materials, and best of all, access to live office hours, 5 days a week, 2 hours at a time. So you can get the one-on-one help that you need for your MCAT prep even through an online course. Again, that's Next Step Test Prep and their MCAT course. Use the promo code MCATPOD to save $50 off their course. If you qualify for FAP, email them, let them know, and they have a very generous deal for you if you qualify for FAP and you need some MCAT prep as well. Again, that's nextsteptestprep.com and use the promo code MCATPOD. That's all capital letters, MCATPOD. Have a great week. We'll see you next time here on the MCAT Podcast.